Hi everyone. I hope I'm audible. Okay, so before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with a bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So for Telegram channel, so join with us in Telegram for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. So let me just check once. Yes, okay. Alright, so for homework and class notes, you can join the channel Vani Kari ICSE Class 9 and 10 Biology. You can search. Just a second. So that is okay. So class 9 and 10 biology you can search, and the Telegram channel of Unacademy, ICSE and ISC both. So this you will find in the description box. So click on the link and you can join the channel there. Okay. So we also conduct special classes on Unacademy platform. So you can learn from your favorite educator for free. So there are special classes which are absolutely free. Okay. So different educators are coming up with different special classes where we will be conducting a quiz also, and you can know your rank with the leaderboard that we will be sharing at the end. Okay, so engage with educators via polls, clear your doubts in the live special class. Okay, so for the top special classes you can visit the link in the description. So now Unacademy subscription features, so you can learn live from the comfort of your home. Unlimited access to all the courses will be there. So once you take the subscription, so different educators are coming up with the different courses, so you will have access to all the courses done by the by the educator. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. Regular doubt clearing session will be there, like after about every 3 session, the 4th session will be a doubt clearing session. So any doubt you have related to the topics that are previously covered, so you can clear your doubt in a doubt clearing class or a session. And also we call it as an answer writing session. So let's say you do not have any doubt, so we can just conduct a quiz, or we can discuss the 3-mark question, 5 marks question. So basically this class will be engaged. So basically we will be covering the important topics in the form of question and answers as well. Then complete syllabus will be covered, mentorship and guidance is given, study material is given. Then along with that there is practice test series, live test series, batch courses. So once you take the subscription, so you will have access to English batch, Hindi batch or a English batch. So you will have access to all of these batches once you take the subscription. And along with that there is a daily practice section, weekly mock test series, live quizzes will be there. So daily MCQ and subjective test series are also given. Okay, so now Unacademy Plus subscription pricing for class 10. Okay, so you can see that you can try with 3 months also. Okay, so for 3 months this is about 2500 per month. So this is gonna vary. So as you go for a higher month, the cost is getting reduced. So for 12 months, so this will come somewhere around 15k. Okay, that is 15k itself, and with the code that is VANIYT, so once you apply the code, so the cost is getting reduced to 13.5k. so you can go for the subscription using the code VANIYT, where you will be getting 10% discount. Okay, so this is, since this is the very beginning of your class 10, so you can go with the 12 months of subscription, or if you just want to try like how exactly this works, you can also go with the 3 months or 6 months. Okay, yeah, so there is an offer right now. So this offer is basically if you are taking a 12-month subscription, so this is applicable for class 9 as well as 10. Okay, so you will be getting 1 month subscription extension. Okay, so this is 1 month. Then for 18 months it is 2 months. So 24 months it is 3 months. Okay, as such 36, 4 and 48 it is 5 months. So if you are planning to take the subscription, so you can do it within this time period, and the prices are actually raising soon. Okay, so if you are planning to take the subscription, so you can go for this offer. Okay, Let's get started with the DNA. Just a second. Yeah. So DNA is the basic genetic material for most living organisms. So where do we find this DNA? Okay, so let's see this. Now every cell, okay, so there is a cell. So we will just draw the cell. So inside this cell you can find, so there is a nucleus. Okay, so there is a nucleus. So now inside this nucleus what you can find? So there is a nucleolus that you can find. Okay, and here itself in the form of a chromatin fiber you can find a DNA. Okay, so that means here you can find a chromatin fiber. So this chromatin fiber is basically, you know, this itself is having a DNA. So there itself you can find a DNA. So now what exactly is the use of this DNA then? So whatever DNA is present here, so this DNA we say that this is going to form a particular body feature like what we all have now. Okay, so that body feature we are getting, that is mainly because the DNA is there. Okay, so that means DNA is there. So DNA is coding for a RNA. Okay, so this is coding for a RNA, and RNA is coding for what? RNA is coding for a protein, and that protein will code for a certain feature, like certain body feature what we are getting. So that is mainly because of the protein that is being coded by this DNA. So that means you can understand that. So whatever body features we have, so now you know, let's say have a wavy hair, or maybe let's say I have a brown eye. So in all these cases the body feature that we have, so this is basically with respect to, one second. Yeah, so this is basically with respect to a DNA which is it. So that DNA is the one which is coding, and that coding is done by this DNA. So that is for a RNA, and then RNA is going to code for a protein. So that is when, so one second. So that is when a particular body feature that we can obtain. Okay, so now nucleus, when we mention, so there is a nuclear envelope. So let's see this part. Just a second, one second. So this is actually moving. Just a second, let me just adjust this. Alright. Okay, so now inside the nucleus itself you can find the DNA that is present. So there is a nuclear envelope. Okay, so what exactly is nuclear envelope now? So now there is the outer membrane as such. So you can find the outer membrane, then you can find there is the inner membrane. So this outer membrane and inner membrane together, we can say that this is now a nuclear envelope. So you can see that this is now a nuclear envelope here. Okay, so this is now a nuclear envelope. So you can see there is a outer membrane and an inner membrane. So this one is nuclear pore. Okay, so this is a nuclear pore. So here inside this you can find a DNA. Alright, so now DNA we will have a look at, or a chromatin fiber. So chromatin is just a thread like mass which is having a DNA and then a protein. So we will see how exactly this DNA and protein is arranged. So let's see the nucleosome model. So I'll just write this. So this one we will see. So that is nucleosome model. Okay, so what exactly is nucleosome model? So you know nucleosome octamer, or you can see there is a histone octamer formation. So this histone octamer, along with that you can find a DNA which is coiled around this. So that we call as a nucleosome model. So let's see this part. I'll just show you this. Okay, so now you know these are the histone proteins. Okay, so which is forming the structure as such. So this is now, this is a histone protein. Okay, so this one is now a histone protein which is forming a bead. So nucleosome model can also be called as a bead on a string model. Okay, so this can also be called as a bead on a string model also. Okay, so this is how such a big DNA is actually arranged into this particular cell. So that is by forming a bead, and then you can find a string. So now this is a nucleosome. So this is a histone protein. Okay, then on the back also you can find the arrangement of histones over there. Okay, so there you can find there is a histone arrangement. So one second, so this is a histone arrangement now. So now this whole thing is basically a histone protein. Okay, so coiled around this histone protein, so you can find the DNA. So now this one we will just write the names of this. So histone protein, when we mention, so there are different types. Okay, so there is H2A type of protein, H2B, H2A, H2B, then H3 type of protein, H4 type of, so these are all histone proteins. Okay, so this is now a histone protein. So on the back also it is H2A, H2B, then you can find a H3 and H4. So coiled around this you can find there is a DNA. So DNA is basically coiled around this structure. So we can just show it like this. So this is now a DNA which is coiled this way. Okay, so this is now one nucleosome. So this is actually a nucleosome, where you can see that the DNA is coiled. So DNA is coiled around what? DNA is coiled around a protein. So this we call protein octamer. So that is a histone protein octamer. So we can just write it. Histone protein octamer will be there. So coiled around that you can see this is now a nucleosome model. So as such you can find many of the nucleosome models. So we will just see this. So let's say this is a DNA now. Okay, so here you can find a bead again. Here you can find a bead. Here you can find a bead. Okay, so this one repetitive units of this. So this is now many units are there. So you can find as such. Okay, so this we can call as a chromatin fiber. So this complete thing with the many histone proteins. So many nucleosome. Okay, so many nucleosome you can find. So not just one. So many of the nucleosomes are there. So that is a chromatin fiber. So now this chromatin fiber which is there, so this is this is coiled and supercoiled. Okay, so you can see the coiling of this particular chromatin fiber. So chromatin fiber which is there, so this is coiling. Okay, so I'll just show you this coiling part now. So now this is a DNA. So there is a chromatin fiber here which is coiled. Then you can also see there is a supercoiling as such. So then there is again coiling, then supercoiling. So this one will go on. So it will continue as such. So there is a coiling. Okay, and then you can see a supercoiling. So this one is basically a supercoiling of a DNA that can be seen. So this is a chromatin fiber which is coiling and supercoiling. So about 2 meters of DNA which is there in each cell. So that is arranged in a nucleus. So 2 meters of DNA that you can find in each of the cell. So this is coiled. So this DNA is completely. So how exactly? Like such a big DNA, like you can imagine 2 meters is a big one. As you compare a tiny cell which you cannot even see with the naked eye. Okay, so it is not even a cell actually. So inside the cell, only in the nucleus, so such a big DNA, about 2 meters of this DNA is completely packed in here. Okay, so how exactly this is possible? Because you can see there is a coiling, there is a supercoiling, and that is how such a big DNA is just compactly arranged here into this tiny place. So that tiny place is nothing but inside the nucleus. So where the nucleoplasm is present, so there you can see this coiled and supercoiled DNA which is present there. Okay, so that is, you can see such a big DNA arranged in this tiny structure. So that is how we can see. Now chromatin fiber, when we mention, so this is coiling. Now chromatin fiber is having many nucleosome models. So this you can see, so this is the nucleosome model. Okay, so you can find many such nucleosome models. So this is only one nucleosome now. That means a core of a histone protein. So now this whole thing, so I'll just make this as a bead. So now this is a bead. Okay, so bead is there. So coiled around this bead you can find a string. So that means DNA is there. So coiled around the DNA you can find this particular protein. So this is a protein and this one is a DNA, and that is why we also call this as a bead on a string model or a nucleosome model. So this whole thing is a nucleosome. Now chromatin fiber, when we mention, so as such you can find many nucleosomes arranged, and all the many nucleosomes itself are coiling here. Okay, so there is a coiling, there is a supercoiling that can occur, and such a big DNA is basically compactly arranged. So that is in a tiny space where you cannot even see a naked cell with the naked eye. So in that case, such a big DNA is basically arranged in that tiny space. So that is because of the coiling, supercoiling, and along with that this nucleosome model is going to help in constricting this DNA content. Is this clear? Any doubt? So this is one thing. Okay, how exactly DNA arrangement that you can see. So this one, I'll just go to this part. So now you can see this is a chromatin fiber now. Okay, so you can see this is labeled as a chromatin fiber. Okay, then you can see chromosomes only when the cell is undergoing for a cell division. Okay, so when the cell is ready to go for the cell division, so that is when you can see that the chromatin fiber will start to condense. Okay, so chromatin fiber will start to condense, so it will lead to the condensation of chromatin fiber, which can form a chromosome. So there is a chromosome formation that you can see. Okay, so now this chromosome which is there, so let's say here is this chromosome. Okay, so this is the chromosome. So you can see that there is a arrangement of chromosome done in here. Okay, so that is inside this nucleus itself, and now this this chromosome is going to undergo many phases, like so there is a karyokinesis, cytokinesis will occur. So under karyokinesis you can see that, so there is prophase, then metaphase, then you can see a anaphase, telophase. So all these phases you can see. So that is because the chromatin fiber is not there, it is not there in the form of chromatin fiber itself. So it will all start to condense. Okay, so that means coiling and supercoiling will occur. So I'll just show you how exactly this occurs. Now you can see this. So this is now a DNA. So DNA is happening what? So DNA is forming a nucleosome. So you can see there is a coiling of this one. So many such coiling you can see. So there is a coiling and supercoiling. So now in that case, now let's say this is a chromatin fiber which is supercoiling. Okay, again there is a coiling, supercoiling. So as such it will continue. So first this is going to form what? This is going to form a chromatid. Okay, so now you can see the chromatid formation you can see. So further you can see coiling and supercoiling there. So in that case we can see this one is further coiling, then you can see the supercoiling, then further coiling, supercoiling. So this will continue as such. Okay, so there is a coiling, there is a supercoiling as such. Okay, so now you see this part. Okay, So where a complete chromosome is being formed. So how exactly chromosome is being formed? So now here I have shown only one. So as such you can see double the content of DNA if it is there. So you can find two arms of this chromosome. Okay, so that means again there is coiling here. So then there is supercoiling here. Okay, so this is going to form a chromosome. So you can see that the chromosome is basically nothing but the DNA that is arranged. Okay, so that is by coiling and supercoiling. So this is going to arrange itself as a chromatid. So now this is a chromatid, this is a chromatid. So on the whole, what is this? So now this is on the whole is a chromosome. Okay, so what exactly is chromosome made of? So this is basically having a chromatin fiber. So chromatin fiber which is actually coiled as well as supercoiled. Okay, so coiled and supercoiled DNA you can find in here. Is this clear about the structure of chromosome? So this is how you can see the coiling. Alright, so next one more thing we will see. Okay, so now we will see the DNA. So now DNA, when we mention, so DNA is basically made up of, so I'll just write the full form of this. Just a second. Okay, so DNA is deoxyribonucleic acid. Okay, so this is what? So this is deoxyribo. Okay, nucleic acid is a DNA, or we can just, this is now a deoxyribo. So this is now a nucleic acid. Okay, so I'll just erase this part 1 second so that. So DNA, when we mention, so by their name we figuring. So now this is a deoxyribose sugar it is. Okay, so this is a deoxyribose sugar. Nucleic acid is this. So this is nucleic acid. Okay, so this is the full form of a DNA. So now what exactly is DNA made up of? So DNA is actually made of polynucleotide chain. Okay, so it is made of what? This one is made of polynucleotide chain. So what exactly is nucleotide or a nucleoside? So let's see that. 1 second. Just a second. Okay. Yeah. What I was saying, so nucleotide, right? Yeah, so nucleotide. So now nucleotide, when we mention, so nucleotide is basically made up of there is 1 second. Yeah, so there is a phosphate group. Okay, so phosphate group is there, and with that you can find, so there is a pentose sugar. Okay, so there you can find a pentose sugar, and along with that you can also find, so there is one more thing. So what is that one more thing? Anybody has any idea what is this? Any of you? No idea? Okay, so this is basically pentose sugar is there, and with that you can find a nitrogenous base. Okay, so there is a nitrogenous base which is going to form a base pairing. Okay, so now this is a nucleotide. So what exactly is nucleoside? So there is one more called nucleoside. So nucleoside also you can find. So this they can be this question can be asked, but anyway for class 10 they won't ask, but for higher classes they can ask. So nucleoside is basically pentose sugar plus a nitrogenous base, so that is without a phosphate group. Now including a phosphate group, we can say that this is a nucleotide. So nucleotide, when we mention, so phosphate group, pentose sugar, nitrogenous base will be there, or otherwise the pentose sugar and nitrogenous base, so it is only the nucleoside. Okay, so let's have a look at this part. where you can see the different nitrogenous bases. So now what are those nitrogenous bases? We will see. Just a second here. There is no space. No. I'll write it here. Okay. Okay. So now nitrogenous bases when we mention. So you can find two types of nitrogenous bases there. Okay. So what are those nitrogenous bases? So nitrogenous bases you can find two types. Just a second. Okay. So there is a purine. Okay. So you can find purines. You can find pyrimidines. So there are purines and then there are pyrimidines that you can find. Now when we mention about a purine, so purines are of two types again. Okay. So there is adenine and a guanine. Okay. So we will just see this. So adenine is there and then there is guanine. And here under pyrimidine, so there are two types again. So one you can find a thymine, okay, and the other one is cytosine. So this is only with respect to the DNA, okay. So this is only in DNA. If it is RNA, okay, so there won't be thymine, so you can find a uracil. So uracil is another nitrogenous base that you can find only in case of RNA instead of a thymine. Okay, so there is no thymine over there. So this is with respect to only DNA. So in DNA you can find a thymine and a cytosine as the nitrogenous bases, and these are the pyrimidines. So let's see the bonding that can occur. So I'll just explain it to you here. So what is that bonding we can see? Okay. So whenever a purine, like purine is always going to make a bond formation. So that is a hydrogen bond with the pyrimidine. Okay. So either this will be with two hydrogen bonds or this can be with the three hydrogen bonds. Okay. So purine is always, so according to the Chargaff's rule, so there is a person who discovered about this one. So Chargaff. Okay. So according to Chargaff's rule, so Chargaff's rule says that the amount of purine is always equal to the amount of pyrimidine. So that means if A is there, so it is always going to pair with the T. So that is by with the two hydrogen bonds. Okay. So this is two hydrogen bonds you can find in here. And the other case, so let's say if there is a G, guanine is there, so this is always going to make a pair with the C. Okay, so that is by three hydrogen bonds. So here you can see three hydrogen bond formation. Okay, so now same way, let's say here T is there, so in that case you can find two hydrogen bond, adenine is here. So let's say here the C is there, so that means again three hydrogen bond and the pairing is gonna be with the G. Okay, so this we call as a Chargaff's rule. So always the purine is always equal to pyrimidine. So that is the number of or amount of purine is always equal to pyrimidine. So that says about the Chargaff's rule. And you have to remember the base pairing that will occur. So that is, this is now called as a base pair. So this is one base pair, that is T is always complementary to A or A is always complementary. So we can use the term complementary as well. Okay, so instead of base pairing always, so we can say that A is complementary to T and G is complementary to C. Okay, so that means G and C are going to form a bond with three hydrogen bonds. T and A are going to form a bond with two hydrogen bonds. Okay, so this is the one, and other than that, just a second, I'll just check. Yeah, so now one nucleosome which might be there. Yes, is this clear? Any doubt till here? Let me just check. Okay, here it is. Okay. Alright, so in this case you can see that one nucleosome. Okay, so now we studied about a nucleosome, right? So one nucleosome may contain about 200 base pairs. Okay, so you can find 200 base pairs of DNA helix. Okay, so now one DNA helix is there. So DNA helix is what? So I'll just show you this. Just a second here itself I can show. Alright, so now you know DNA helix is this one. So you can find a DNA structure. So you can see the coiling of a DNA as such. So now this is a DNA double helix. Okay, so this one is DNA double helix. Okay, so now in DNA double helix you will find one of the DNA strand running in 5' to 3' direction, the other DNA strand running in 5' to 3' direction. Okay, so that means now both of these DNA strands which are there, so DNA strands are anti-parallel to each other. So why do we say that this is anti-parallel? So let's see, now this is the DNA strand which is in this direction, like you know from 5' direction till the 3' direction. Okay, so this one is 5' to 3'. So here I have written this one second, let me just change it. Yeah, so this is supposed to be 3' and the other one is supposed to be 5'. Okay, so in that case this is 5'. So here you can see one of the strand running in 5' to 3' direction, the other one being, so you can see this one, the other one is running in 3' to 5' direction. So that means they are not parallel to each other, but instead they are anti-parallel to each other. So when do we say that they are anti-parallel? So let's say this is a DNA strand now. So one of the DNA strand goes this way, the other DNA strand is going this way. So this we call as a anti-parallel nature. Okay, so going in this direction means, so let's say this is 5' direction, the other one is 3' direction. So here it is 3' direction and here it is 5' direction. So I'll tell you what exactly is this one. Okay, so let me just clear this up and explain it to you. So why do we say that it is 5' or 5' or why 3' or 3'? Okay, one second. So now here in this case, let's see this. Okay. Okay, so we'll start with the 5' or maybe I'll just show you this one. So now this is a structure of a DNA. Okay, so this is a structure of a DNA. So we will just draw the deoxyribose sugar. Okay, what I'm drawing now? So I'm drawing a deoxyribose sugar. So in that case you can find HOH. So this is basically a nitrogenous base you can find here. So this is going to be nitrogenous base. Okay, and here you can find H. So here again you can find HH. Okay, so here it is HOH and this is going to be H, and this side, so this is CH2OH. So this is the structure. So this is what? This is a deoxyribose sugar. So this one we will just draw. So this is now a deoxyribose sugar it is. Okay, so deoxyribose sugar, so this one is a nitrogenous base. Okay, then attached to this one you can find a phosphate group. Okay, so this one is completely what? This is now a nucleotide. So this we call as a nucleotide because it is having a phosphate group. So it is having a nitrogenous base and it is having this deoxyribose sugar. So now you can see that this is the first carbon. Okay, so this is first carbon, this is second carbon, this is third one, this is fourth carbon here. Okay, and here you can find a fifth carbon. So pentose sugar, so we also call this sugar as a pentose sugar as well. So what is the meaning of pentose sugar? So that means 5 carbon groups you can find. So now over attached to this one, you can find one more sugar molecule in conjugation. So we will just erase this part. So I'll just show you here. 1 second. So I'll just erase this part. Okay. Okay, so here the nitrogenous base is gonna get attached. So we will see this. There is one more sugar here. Okay, so there is one more sugar. So same as that of a deoxyribose sugar. Okay, so now again as usual you can find a nitrogenous base. Then you can find HH. Here it is H and OH. The other one is H. Then you can find a CH2. Okay, so this is CH2OH. So this is basically forming a bond with the phosphate. Okay, so this is going to be OH. So in that case, so this is CH2O and here this is a phosphate group. Now this phosphate is going to make a bond with the third one. So you can see that this is the 5th carbon. So phosphate group which is making a bond with the next nucleotide here, so that is free at this part because here it is not making a bond with any other. Okay, so I'll just show you this. Okay, 1 second. Yeah. So here it is not making a bond with any other because on top you cannot find any nucleotide. So now this is a 5' or 5'. The other one you can see that it is making a bond with the 3rd carbon of the previous nucleotide. So we will draw one more and see. Okay, so now one more nucleotide is here. So this is another nucleotide. Okay, so that is of the DNA. So as usual nitrogenous base you can find. Then there is HH. So this is going to be HOH. Then you can find H and here it is CH2. Okay, so this is CH2. OH. Okay, so that means here a phosphate group is there. So now one of the phosphate group is making a bond with the 5th carbon and the other one is making a bond with the 3rd carbon, and that is how we say this as a polynucleotide chain. So DNA when we mention, so this is made up of many nucleotides, so that is why we say that this is a polynucleotide chain. Okay, so now this one is a DNA. All right, so this is a DNA now, and you can see that at the 3rd position, so OH group is free, so it is not reacting with anything. So we can say that this one is a 3'. Okay, so this we call this as a 3'. So 5' to 3' when we mention, so that means the DNA is running in 5' to 3' direction. So that is mainly because the 5' end is free here and on the other side the 3' end is free. So this we call as a polynucleotide chain. Okay, so now we mentioned that the other strand is running in 3' to 5' direction. So that is 3' to 5' we say. So that means it is anti-parallel in nature. So we will see how exactly this is 3' to 5'. Okay, so we say this as an anti-parallel nature. So now you know we need to make these nitrogenous bases coming together so that they can make a bond with the next strand, that is next DNA strand which is here. So in that case let's draw this. So here you can see, so this is gonna run in a reverse way. Okay, so in that case you can see this one being the nitrogenous base. So here being the nitrogenous base, so you can find a nitrogenous base here, so that is at the 1st carbon. Then you can find 2nd carbon, so 2nd carbon being H and H, the 3rd carbon being OH and H, and here you can find a CH2OH. Okay, so CH2OH and phosphate group is this. So this is as usual H. So one more nucleotide we can draw. Okay, so that means here a sugar you can find. So this is now a sugar. Just a second. Okay, so now this is a sugar now as usual. So you can find a nitrogenous base there. and here you can find H and H. So here you can find H and here you can find OH. So now you know this is going to form a bond. So you can see that. So two of the bond formation. So phosphodiester bond. So phosphate is making a ester bond. Okay, so ester bond here, ester bond here. So that is by losing a water. Okay, so phosphodiester bond formation we can see in the DNA backbone. So on the other side also is the same thing. So now we will see one more. That is sugar, that is nucleotide. Now so as usual, so here CH2 is there and P is here. Okay, so there is a phosphate group and here this is a nitrogenous base. So now you know this is starting from 3 prime. Okay, so why do we say that this is 3 prime? Because the third group is ready to react here, so it is free. And here the fifth one, so because the phosphate has not made a bond with anything, so here the fifth prime or fifth group is free to react with the next nucleotide. So in that case this is going to be same. So as usual the HOH, so this is HOH and here H. So this is, you know, this itself we call as a complementary DNA strand. So depending on the type of base pair, so either they are going to be, so let's say this base pair is now, since I have shown 3 bonds there, so let's say this is G and C. So this is A and T. So we can also show this as T and A. So these are the nitrogenous base. So A is complementary to T, okay, and G is complementary to C. So T is complementary to A or C is complementary to G. So that is how the arrangement of these DNA strands we can see. Okay, so this is now a anti-parallel nature, and that is why we say that the DNA is anti-parallel and complementary to each other. So now one nucleosome may contain about 200 base pairs of a DNA helix. So now this is only one base pair. So what I have shown here, so this is only one base pair. Okay, so as such you can find how many base pairs? 200 base pairs you can find. So that is for only one nucleosome. So what is nucleosome? So nucleosome bead is there. Okay, so coiling that nucleosome bead, you can see that the DNA is coiled around this one, forming this type of a structure. So this we call as a nucleosome. So you can also show it this way, or let's say this is a nucleosome protein, that is a histone protein, and you can also show that the DNA is coiled this way as well. Okay, so this is also possible. So you can go for any of this. So nucleosomes repeat in a thread-like structure, so that is called as a chromatin, which is present in the nucleus. So you can see that the nucleus itself is having these many of the nucleosome. So that is about 2 meters of a DNA is arranged in here. So I will just show you this. So you can have a look at this question. So what is the total length of a DNA that a human body may have? Any idea about this? So how many, like, you know, what is the total length of a DNA that a human body may have? So if one cell, okay, so it is approximately said that one cell is going to have about 2.2 meter of DNA. Okay, so this is only for one cell. Now as such we have trillions of cells, not just 1 or 2 cells. So trillions of cells you can find in a DNA. Okay, I mean so cells you can find a human body, not a DNA. So that means 2.2 meter into trillions of cells if we are considering. So anyway, that is approximate range I can show you. So approximately this is going to be about 110 billion miles. Okay, so if one DNA strand is about 2.2 meter, okay, so that is the DNA from the 100 trillion cells. You can see trillions of cells there. So from 100 trillions of the cells, if we are putting them end to end, so let's say we are joining all the 2.2 meters of DNA end to end, so this would take us to the sun and back about 600 times. So we would reach the sun. So with this approximate DNA that we have, in our body, like about 100 trillion cells are there if we say. Okay, so if we are joining all this DNA together, like let's say a 2.2 meter, we are just joining them end to end, so we would go back to sun, like we can go to sun and come back to earth, so that is about 600 times. So such a huge amount of DNA is there, and that is basically how exactly this is arranged. Such a huge DNA is arranged in our body, so that is by coiling. Okay, and then supercoiling. So coiling and supercoiling we can see. So basically this is going to form a chromosome as such. So now DNA is arranged in the form of a chromosome, so that is by coiling and supercoiling. So this is only when the cell division has to occur, so you can see the chromosomes clearly. Okay, or otherwise you cannot see chromosomes, so you can just see this DNA as a thread like structure normally. Okay, so here they have shown in this picture. So next rest stop 30 million miles. So in this case you can see this is the DNA now, where they are showing a base pair, so it is used as a ladder here. So with that you can see that, so we can reach the sun about 600 times, so only with the body DNA which is there. Is this clear? Any doubt till here? So this is about the approximate length of a DNA that a human body may have. Just a second, I'll drink some water. Okay, all right. Yeah, so I was discussing about this. So nucleosome, when we mentioned, so nucleosome is basically that coiled DNA itself. Okay, so that means coiled DNA is there. Okay, so chromatin fiber, so in a nucleosome, or if we are saying about a chromatin fiber, so one nucleosome is going to have about 200 base pairs. So what exactly is nucleosome? So this is a histone protein. So coiled around that histone protein you can find a DNA. So let me just change the color so you can understand which is DNA here. So now DNA is coiled around this histone protein. So this is now a DNA. So now this whole thing is actually a nucleosome model. So you can find about 200 base pairs of a DNA in this, only in this particular DNA. So as such we can find many nucleosome repeats. Okay, so that is in a thread like structure, so that we call as a chromatin, which is present in the nucleus. Okay, so you can see further supercoiling of this chromatin, which creates a loop structure of chromatin fibers. So you can see that the chromatin fiber formation. So I'll just show you this, how exactly this occurs. So let's say this is now a chromosome. Okay, so in that case, now chromatin fiber. So this is coiling, then this is supercoiling, again there is coiling, so you can see supercoiling. So this way this will go on coiling. So first it is going to form a chromatid. Okay, so this one is a chromatid now. So there is a constriction here. So as such, again there is, this is going to coil, this is going to supercoil, again there is coiling, supercoiling. So this is going to take this type of a shape now. Okay, so this shape you can see, so that is mainly because of the arrangement of the chromatin fiber. So this thread like structure called as a chromatin, which is present in the nucleus, is giving this structure. Okay, so further supercoiling, chromatid will be there, and then this is further condensing. So then you can see a chromosome structure. So this is just single arm I have drawn. So in S phase what happens? In S phase the DNA is actually getting doubled. Okay, so DNA replication you can see in S phase. So in that case you can find double the amount of DNA. Okay, so if the double the amount of DNA is there, so we can just show one more arm as such. So there is coiling and then there is supercoiling. So there is a sister chromatid formation that we can see. Okay, So sister chromatids are formed, and here we can see that this is a chromosome. So this is coiled and supercoiled. So DNA is basically double the amount here. Okay, so double the amount of DNA you can find. So that is mainly with respect to the DNA, I mean, the chromosomal arrangement. So now this is a chromosome. Okay. Any doubt till here? So we will go to this question section. Any doubt? Any of you? Okay. Okay. So where is DNA present in the eukaryotic cells? So where exactly do you find a DNA? Okay, so that is in a eukaryotic cell. So that is inside the nucleus with other cellular content, inside the ribosome and not present. So where exactly do you find the DNA? So that is in a eukaryotic cell. Any idea? Yes, no idea. Where do you find the DNA? So DNA is present in eukaryotic cells. So they are asking about the eukaryotic cell, not the prokaryotic one. So what is eukaryotic cell? That means well-defined nucleus you can find. Okay. So there is a well-defined nucleus. In case of prokaryote, what do we see? The nucleus is a primitive type of a nucleus. So that means the nucleus is not well-defined. Okay. So here it is well. Okay, one second. So the nucleus is actually well defined in this case. So that means DNA you can find inside the nucleus itself. Okay. Now when we mention about a prokaryotic, so randomly this DNA is present in the cytoplasm, and wherever a DNA is present, so that space we can call as a nucleoid. Okay, so that we can call as a nucleoid. So that is with respect to prokaryotic cell. Here in case of eukaryotic cell, the DNA is present inside the nucleus. Okay, so let's see this one now. Which of the following comes under the level of organization of DNA? Okay, so which is that level of organization of DNA? So nucleosome, chromatin, fiber, solenoid, and all of this. Any idea about this? So this is basically DNA itself which is coiling. Then there is supercoiling. So that means you can see that this is a solenoid. What is solenoid? Solenoid, when we mention solenoid, okay. So when we mention, so this is the one. So this you will study in physics, right? So that is coiling has occurred. Then this itself is supercoiling. So coiling, supercoiling. So there is a chromatid formation. Then there is a constriction here. So chromatin fiber is arranging itself as a solenoid. So chromatin fiber is made of a nucleosome. So the answer is all of the above. Okay. So now in the beads on a string model, so that is basically a nucleosome model. Okay. So the bead is made up of 6 histone protein, 8 histone proteins, 6 histone proteins, and then the DNA. 8 histone proteins and then the DNA. So what is the case here? So now you say that this is a bead, okay, or this is basically a protein bead or a histone protein bead we can say. So this is basically a histone bead, okay. So now you know what it is, how many beads of these are there. So you can find 8 histone proteins in that. So coiled around that 8 of the histone protein you can find, so there is this DNA which is coiled. So this is now a DNA. So this is now a histone protein. So we can say that 8 histone proteins along with the DNA you can find in a nucleosome model. Okay. So this is the nucleosome model itself. So single human chromosome may have about million nucleosomes. Okay, so you can see this is a phone wire. Okay, so this one is actually a telephone wire, where you can see there is a coiling, then you can see there is a supercoiling. Okay, then again you can see coiling and supercoiling. So this is how the chromosome is arranged. Like chromosome is condensing, so that is by DNA chromatin fiber goes on condensing, and it will form this type of structure, so that we call as a chromosome. So chromosome is basically coiled and supercoiled DNA or chromatin fiber you can find. Okay, so DNA packaging in case of eukaryotes, so this I already said, like how exactly this is present. So proteins in eukaryotes, so this one is basically the protein is positively charged. Okay, so this protein bead, what is there? So this is positively charged, so that is because of protein. When we mention it is made of a amino acid. Okay, so there is a amino acid called as arginine and lysine. Okay, so they are providing, so there are amino acids like arginine and then there is lysine. So these are the amino acids. Okay, so these amino acids are more here, which will give this protein a positive charge. Now the DNA which is coiled around this protein, so this DNA is negatively charged. What is the reason for DNA being negative charge? So that is because of the phosphoric acid or the phosphate group which is there, so that is providing a negative charge. So positive and negative charges, so they are making this bond to remain stronger. So there is a positive and negative attraction that will occur between a protein and a DNA, and that is how this particular, so this is actually positive charge. I'll just change this. So this is supposed to be positive, so that is because of the arginine and lysine that you can find. Okay, so there is a arginine and then there is a lysine which are providing a positive charge. So this positive charged protein and the negative charged DNA, so this negative charged DNA is there, so that is the reason why there is a opposite charge attraction is there, and that is how the DNA and the protein are compactly held with each other. And then you can see this nucleosome arranging, like many nucleosome formation you can see, and there is a chromatin fiber. Okay, so now amino acids are lysine and arginine type of amino acid which are forming a histone, and they are the ones which are providing a particular charge, so that is a positive charge. Now 8 molecules you can find. So what is that 8 molecule? Histone proteins which are there. So this is now a histone protein. Okay, so which is forming one complete bead, just like a ball. Okay, so ball is there, so surrounding that ball you can find the DNA which is coiled around. So it is same as that. So histone octamer you can find. So now on the back side you can find another set of histone protein. Okay, so based on that, so there is a ball formation, so octamer. So here you can find H2A type of protein, H2B, then you can find H3 type of protein, H4. So this is now one histone bead. Okay, so bead on a string model, then coiling around this one you can find there is a DNA. So this is DNA coiled around this one. So in that case, now on the back side, so this is the back side now. Okay, so on the back also you can find, so there is a H2A, H2B, H3 and a H4 type of histone protein. So 8 of the histone molecules or histone protein molecules which are positively charged. So that is mainly because there is arginine and lysine type of amino acids are there. So they are forming this octamer here. So all the histone protein. Now with the negatively charged DNA is going to wrap around this protein, forming the nucleosome model. So repeated units of a nucleosome. So I'll just show you this. Repeated units of a nucleosome. So let's see. Here one nucleosome is there. Here one. Then you can find here. Okay. So now repeated units of nucleosome when we mention. So this we call as a chromatin fiber. So this is now a chromatin fiber. So there is a chromatin fiber. So many repeat units. So this is basically nucleosome repeats. Okay. So nucleosome repeats itself is a chromatin fiber. So this chromatin fiber itself. So this whole thing is a chromatin fiber. So this itself is coiling like this. This itself is supercoiling like this. And this is going to form a chromatid. So now again this is going to coil like this, supercoiling, and then it is going to form a chromatid. So you can see that whole chromosome is formed now. So chromosome formation is nothing but the chromosome is made up of chromatin fiber itself, and chromatin fiber, when we mention, it is having a many nucleosome repeat, and this is one nucleosome now. So that is how the DNA is arranged in this tiny nucleus that you can see. Okay. Any doubt in this? Any doubt that you have? Hi Darsh, good evening. Any doubt if you have, you can ask. So name the scientist who proposed the nucleosome model. Any idea who's the scientist who proposed the nucleosome model? So this is the Roger D. Kornberg. Okay. So this Nobel Prize winner American scientist showed that the nucleosome serves as the general gene repressor and prevents the initiation of transcription. That means, so DNA full form. So DNA full form is deoxyribonucleic acid. So I'll just write it here. So this is deoxyribonucleic acid. Okay. So this is DNA. All right. Yeah. So now Don and Ada Ollens were the first to discover the structure of a nucleosome. So now who exactly discovered the nucleosome. So it is the Don and the Ada Ollens. But the further study related to this nucleosome was done by the Roger Kornberg, and he said that whenever there is no requirement of RNA, so basically DNA, from DNA itself RNA is being formed. Then from RNA itself protein is being formed. Then the protein is the one which can give you a particular body expression, like a body feature we can say. Okay. So body feature, like you can have a brown eye, you can have a curly hair, you can have a straight hair, you can have a different complexion. So all these features, like body features that we are getting, so that is mainly because DNA is coding for a RNA, and RNA is coding for a protein, and protein is coding for a particular body feature. So based on that, we are all having a particular feature. So now DNA or a nucleosome model. So this was first given by the further study related to nucleosome was done by Roger Kornberg, but the discovery related to nucleosome was done by Don and Ada Ollens. Okay. All right. So before I end this session, okay, so let me just say some of the features of Unacademy. So now we are conducting free classes, that is ICSE Premier League. Okay. So series of free sessions to cover important topics and questions with Unacademy top educators. Okay. So here you can attend these sessions. So PCMB subjects are being covered. So this is going to start, like this has already started on 6th of May and it is till 31st of May. All you need to do is download the Unacademy Learner app. Okay, so download the Unacademy Learner app and start attending the sessions. Like basically we will be covering, all the different educators are going to cover the important topics and questions. So basically we will be discussing 3-mark question or 5 marks question in these sessions, so that this is going to be helpful. So these are all taken from, like previous year questions are taken, or maybe we will take them from the reference book. So you can go for any of this. So just you need to download the Unacademy Learner app and start attending the session. So these classes are anyway, so these are free classes which we call as a special classes. Okay, and this is now a batch. So there is a gladiators batch which started on 4th of May, but yes, only 3 classes are done. So you have an option to watch the recordings as well. So once you take the subscription, so you can attend the live classes. So there are many more classes which are yet to go live, so you can take up those session. Yes, so this one, the premium IPL is basically free of. This is free class. Okay, so these batches are anyway plus batches, I mean plus classes. So for this you need a subscription. Okay, so here the mode of delivery is English, and the other part, so this is going to start from May 11th. So here you can attend the live session. So in that case